Alright guys, so we're going to start off this new unit with a little bit of history about DNA. DNA is the most important molecule in our bodies, essentially. It's important for carrying the genetic information that is passed on from generation to generation. And it actually has a really interesting history and a ton of people who are involved in its overall discovery and different properties, which we'll talk about when we talk about the overall structure of DNA and the replication of DNA. So we're going to start off with some of the older experiments, and these two experiments are very similar in what their purpose was. So back in about 1927, there was this guy named Griffith who set up a very simple experiment where he took virulent and non-virulent strains of a bacteria and injected them into live mice, and he wanted to see what would happen. So here you can see these different strains of this bacterium here, and what he noticed was that any mice that was inoculated or given the strain that was living, that was virulent, would die. And then he noticed the ones that were non-virulent, or the mice that were given the non-virulent strains or the killed strains, lived. So he was the first person to kind of coin this idea of bacterial transformation. So what that means is that healthy cells taking in DNA or something from these bacterial cells. At the time, he didn't really know what caused that. DNA wasn't discovered yet. But he was just really the first one who coined bacterial transformation. In about 1944, so a little less than 20 years later, these three guys, Avery, McLeod, and McCarty, they developed an experiment to test was DNA actually the transformation factor? So was it actually DNA from those bacterial cells that were going into the normal cells causing the mouse to die? And they were the first ones to provide experimental evidence that DNA was the transformation factor. In about 1950, this idea of DNA had kind of taken off, and this guy, Erwin Chargaff, did a couple of experiments where he compared DNA samples from different organisms. And again, at this point, we didn't know what it looked like yet. But what he did find were that there were these nitrogen bases that were involved in DNA in all of these different animals that he was looking at. He noticed something that are called purines, which are double ring structured, and he noticed that there were pyrimidines, which are single ring structures. After careful analysis of these different DNA samples, he found that in DNA, all of the A's plus the G's would equal the number of T's and C's. More specifically, the number of A's and T's would always be equal, and the numbers of G's and C's would always be equal, because these two structures bind together, so A to T, G to C. We'll talk about that a little bit more with replication, but the overall structure of DNA would not have been able to be discovered without Chargaff's rule here, saying that the A's plus the G's will always equal the T's plus the C's. So in about 1952, these two guys, Hershey and Chase, came along, and the idea had been floating around about DNA, but no one really knew whether or not proteins or DNA was responsible for carrying the information. And so these guys developed a very simple experiment to test which one was the genetic material. So they used specifically bacteriophages, which are viruses that only infect bacterial cells. So in this diagram, you have these bacterial cells. They coated the outside of the protein capsule of the bacteriophage using a radioactive sulfur. And then on another bacteriophage, they labeled the DNA inside of it with a phosphorus radioactive material. After allowing the bacteriophage to infect the bacterial cells and blending it using a centrifuge, or blending it and then using a centrifuge, they measured where was the radioactivity. Well, in this experiment, they found that the radioactivity was not inside the bacterial cells. So that means the protein did not enter the cell. But in this experiment, they found that the DNA did enter the cell, and that's where the radioactivity was. Therefore, they kind of provided more evidence that DNA was the genetic material. Not only was DNA the genetic material, they also kind of supported the idea of Avery, McLeod, and McCarty experiment earlier. Now we're getting to the interesting part of DNA's history. Rosalind Franklin, it's a name you've probably heard before. Between about 1950s and 1953, she was working with a scientist named Maurice Wilkins. And Maurice Wilkins was kind of like her mentor. And she, a little bit closer to the 1953 range, she actually took this picture, which was, it's known as picture 51. And this picture was taken using a process called X-ray crystallography. And you might not be able to see much from the diagram, but what Maurice Wilkins and Rosalind Franklin discovered from doing this was the overall structure of DNA. They determined that this DNA structure must have been a double helix structure based on these patterns that you see. Unfortunately, with this type of experiment or crystallography in this time period, she was exposed to a lot of radiation. And so she eventually died before she can kind of put her word in. And so to tie it back to Rosalind Franklin, before she could put her word in that she's the one who discovered the overall structure of DNA, at the same time she was working on publishing her work, Watson and Crick came along and they actually stole her work. And in 1953, they published the overall structure of DNA in Nature magazine, or journal, I should say, entitled A Structure for Deoxyribose Nucleic Acid. They talked about the overall structure. They incorporated Rosalind Franklin's picture 51, and by the time the Nobel Prize was awarded, Rosalind Franklin had died, and Watson and Crick took all of the credit for discovering the structure of DNA. We're actually going to spend a little bit of time reading this article and going through what some of the ideas were that Watson and Crick were going over. So, past all of that messy history of stealing people's work and winning Nobel Prizes, we have in 1958, about 5 years later, Meselson and Stahl came along, and they did a little bit more research into DNA replication. And what they found was that DNA replicated semi-conservatively. I'm not concerned with knowing how they did this experiment, but the semi-conservatively part is what's important. So DNA is normally this double helix structure. During replication, that DNA molecule is split apart, and each of those, what's called parent strands, serve as a template for a new strand. So at the very end of replication, where you have these two new DNA molecules, you can see that it contains half of the original and half new strands. And so these guys were the first guys to experimentally prove that.